Okay, the small molecule folate, which we receive in dietary sources, is a very important part of metabolism, and the primary function is going to be involved in what are called single carbon transfers. These single carbon transfers are required in a number of biochemical reactions in order to transfer a single carbon rather than a group of carbon to modify a substrate. So in our diet, we are going to start with folate. Folate gets its name from foliage, which is present mostly in green leaves of plants. Now the folate molecule itself we take up into our bodies, and it's not useful until we perform two series of reduction reactions. The enzyme performing these reactions is called dihydrofolate reductase, and the product of this reaction is tetrahydrofolate. Now, being a reductive process, we are going to use NADPH for these reductions to generate the tetrahydrofolate molecule, which is typically abbreviated FH4 for the fully reduced form. Now, once we have our tetrahydrofolate, we have the molecule that we can now process in order to begin our one-carbon metabolism. So tetrahydrofolate we have to prepare by donating the one carbon. And the one carbon is going to be donated from the amino acid serine to form the amino acid glycine. So the one carbon is coming from the side chain of serine. What we form is a structure that I will abbreviate because this is only a portion of the molecule. Is N5, N10, methylene tetrahydrofolate. So the one carbon that was transferred from the side chain of serine onto tetrahydrofolate, this is just a small portion of the tetrahydrofolate molecule, but it's the business end. So this nitrogen is number 5 in the ring structures. This nitrogen is number 10. So the carbon donated by serine forms a bond between these two nitrogens, and this becomes N5, N10, methylene tetrahydrofolate. Now this particular group, this structure, N5, N10, methylene tetrahydrofolate, can be used for single carbon transfers. This process of methylene can also be oxidized to form the N10 formal tetrahydrofolate. This structure will look like this. Here we have broken the bond between the carbon and the number 5 nitrogen, retained the bond on number 10, and have oxidized the CH2 group into a formal group with a carbonyl in it. This molecule can also perform single transfers. Now the reason that these two molecules can perform these single transfers of carbon is because the bonds between this carbon and the substrate are very weak. We have a carbon bond to two nitrogens here. That makes this carbon a good donor for single carbon transfers. And likewise, the bond of this formal group on this nitrogen here is also a good donor because of the weakness of this bond between the carbon and the nitrogen, because this is a carbonyl. Now this methylene tetrahydrofolate can also be reduced, and when it is reduced, it is forming this structure. This structure is called N5 because the carbon is now on the number 5 nitrogen, N5 methyl tetrahydrofolate. Now notice this bond here is very stable between the methyl group and the nitrogen. So this is not a good donor of carbon because this bond is too stable. The tendency is toward reduction. So if the methylene and the formal directions move and migrate to the methyl form eventually over time, we will have what is called a methyl trap. This methyl trap gets its name from the fact that this methyl group is stable on the nitrogen, and we cannot use this for a one carbon transfer. If we cannot use this for a one carbon transfer, eventually all of the tetrahydrofolate in our body will be reduced into the N5 methyl form, and we will not have any substrates available for single carbon transfer. So hence everything gets trapped into the methyl form. Now the question is, how do we resolve this? Because this is not something that is going to be happening. We have a system in order to alleviate this problem. And this system comes in the form of the formation of S-adenosyl methionine. Now S-adenosyl methionine is a major single carbon donor, abbreviated SAM. This is a major single carbon donor in many biochemical reactions, so we must prepare this molecule, and we are going to do so by breaking down the N5 methyl tetrahydrofolate molecule and using this carbon in a reaction to charge up S-adenosyl methionine and then returning this to tetrahydrofolate. So the reaction we need to talk about is, first we need to start out with the substrate homocysteine. Homocysteine is an amino acid. It is not one of our typical 20 amino acids that is used for protein synthesis, but it is important in this particular reaction. So the first step we're going to perform is to convert the homocysteine into the amino acid methionine. This is methionine. Now notice on homocysteine we have a free half sulfur group here. On methionine we now have a methyl group attached to this sulfur. This is a one carbon transfer to go from homocysteine to methionine. Now we are going to be using the N5 methyl tetrahydrofolate for this reaction. But recall that this methyl is too stable to be a donor. So we have to donate this in a separate reaction to vitamin B12. In doing so, we restore tetrahydrofolate so it can go back to the beginning, receiving the single carbon from serine, and start the cycle all over again. Now in this reaction, the methyl group on N5-methyl tetrahydrofolate is being transferred to vitamin B12. Vitamin B12 is an excellent recipient of this methyl group, and using vitamin B12 we are able to have the energy to remove this stable methyl group from the nitrogen. The vitamin B12 now will be used in the conversion of homocysteine to cysteine by transferring that single methyl group, single carbon, to homocysteine's free sulfur, forming methionine. The vitamin B12 can then go back and continue processing methyl groups on N5-methyl, and therefore we can alleviate the methyl trap problem. Now once we form methionine, we have to continue, because this is a process of building a molecule that can be one carbon transfer donor. And so in this case we are going to take the methionine and modify it with adenine. The adenine we will add to this group. We are actually adding this in the form of adenosine. So this is the nucleotide base. This is the base, adenosine, with the ribose attached to it, making it adenosine. Now what this does is create the molecule S-adenosyl methionine, or abbreviated SAM. S-adenosyl means that we have the adenosine residue on the sulfur group. Methionine is our backbone amino acid for this transfer. Now the important point here is that we are creating 3 bonds on the sulfur. Recall that sulfur likes 2 bonds. So this is creating an unstable sulfonium ion. The least stable bond of these 3 is the bond of the sulfur to the methyl group. This destabilization of the sulfur-methyl bond is what allows the S-adenosyl methionine to be a good donor of carbon. So then the next step is using SAM in the reactions that require single carbon transfer, and in doing so we are regenerating an intermediate in this process of S-adenosyl, because we still have the adenine and the sulfur, but in this case this is called homocysteine. In doing this, we have removed our methyl group for our 1 carbon transfer addition, and then the next step, the final step, is to regenerate homocysteine by removing adenosine from the S-adenosyl homocysteine molecule. Now in doing this reaction, we're able to keep the cycle going for 2 very important reasons. Number 1, we are generating an excellent donor of single carbons in the methyl form from SAM to perform reactions. And second of all, we are alleviating the methyl trap by processing N,5-methyl tetrahydrofolate using B12. So recall when we talked about megablastic anemia, that is typically a folate and or B12 deficiency. And the requirement here is the single carbon transfers are required for DNA replication activity and red blood cell maturation. If we are deficient in folate, we are deficient in regenerating the S-adenosylmethionine, and so our one-carbon transfers are inhibited, and therefore our red blood cells do not properly mature, and they remain very large, leading to megablastic anemia. This can also be mimicked by B12, vitamin B12 deficiency, because if we do not have B12, all of our tetrahydrofolate will end up in the N,5-methyl form, and we will mimic a folate deficiency. Now, vitamin B12 is long-lived in the body and typically not anything that is of deficiency. However, when taking folate supplements, it's often wise to take some B12 supplement also.